Um, and let's put this into the context of what we're going to be doing for our long-term project. So we are going to isolate bacteriophages. We are going to isolate their genomes, and then what? What are we doing with those genomes? We're going to sequence them, is the simple answer. Okay? How do we sequence DNA? These days, yes. So there are many different techniques. Okay? Go ahead, Jonah.
Dideoxy.
That's known as, so that technique is known as Sanger sequencing. That is probably the first, you could call that generation 1 in terms of sequencing techniques. Still widely used if all you're trying to do is analyze a few hundred base pairs. But if you're trying to analyze genomes, we need, actually that kind of sequencing was done on the first human genome, and it took like 15 years to sequence it. All right? Well, by the time that they were done sequencing, new techniques were already available. And now we're at a point where you can sequence a human genome in a week and for $1,000. Whereas that original genome was in the millions to sequence. Okay? So we've come a long way. So what allowed us to speed this up? I'm not going to go through all the different techniques. There's a lot of different ones that are based on different types of chemistry, but they all have some things in common. All use light or fluorescence as a readout for detection, and they're all occurring. Also in this technique, everything's occurring in real time. In other words, as the amplification or sequencing event is going on, you're measuring each nucleotide that's being added within milliseconds, within microseconds. This technique that we're going to use is known as PacBio, or SMRT. PacBio is the company, Pacific Biosciences, and it's one of 3 options that UW has available at their biotech center for sequencing. I'm not worried about you guys knowing all the details, but probably enough so that you could explain this to, say, a board of trustee member at Edgewood Engage. Okay. Or Francie, one or the other. All right, so here's how the technique works. Basically, it's like amplification of single-stranded DNA. And in this technique, what you have, or what this image is trying to show you, is you have this aluminum plate with a bunch of small micropores or microholes. Okay? You can do about 3-4 million of these sequencing reactions on a single little chip. So you take your DNA, you shear it into smaller fragment sizes. Typically, I think the sizes we're going to get are about 3 to 5 kb are going to be sequenced. And what you have at the bottom of the well is basically polymerase physically bound. So DNA polymerase physically bound to the bottom of the well. And it's bound to your fragment of DNA. Each single nucleotide in this experiment is attached to some sort of fluorophore or some sort of fluorescent marker. So C's or cytosines would be tagged with, for example, cyan fluorescent protein, or something that gives off a blue light. It wouldn't be a protein, sorry, it would just be something that will turn blue when you can detect it. G's or guanines might be tagged with something that fluoresces red. Thymines might be tagged with something that fluoresces green, and then you might have adenines bound to something that would fluoresce like a yellow color. Right? So you can detect all 4 of them individually. And then what do? Well, in each of these wells, you basically have a camera that can capture the light. And when light is, or when the nucleotide is incorporated, what you get is this bright signal for a short period of time. It gets clipped off by the polymerase because this is attached to the triphosphates, and then that signal goes away. All right, so in this example here, what we have is they're describing it as a C-pulse. Cytosine comes in, is added, gives off light for a brief moment of time, and then that signal goes away. And then the next nucleotide gets added, and it's an adenine. Get a pulse for a brief second of time, and then it goes away. And you keep getting this over and over again. So if you were to watch, actually, this video here, the comments, somebody describes this as like a molecular party or a molecular rave, because the video shows like all these flashing colors, and yeah, anyway, it's awesome. So, take a look at it, all right? Take a look at the video. It's not too bad.
So the light goes away when the polymerase attaches?
Yeah, so once the polymerase cuts that nucleotide, to add that nucleotide and get rid of this triphosphate here with this fluorophore on it, you'll get this pulse for a very brief period of time, and then it kind of like diffuses out of the well.
Okay.
Because now it's cleaved and gone.
Okay.
Sabrina.
So the pyrophosphate has the like fluorescence attached, so
Yeah.
when you use that
Yeah.
okay.
Now some other techniques are based on detecting that pyrophosphate, but that's not this one. I wanted to point that out. Jonah.
So if all of these little segments of DNA are chopped up from
Yeah.
chromosome, how do you put those in order and figure out?
We're not going to do that.
Okay.
But that's a great question. They asked me if I wanted to, and I was like, yeah, sure. And then I realized how much time we had, and I was like, yeah, no, we're not going to do this. So, really what you have, imagine you have a phage genome that's about, let's say, 80,000 kb, all right? If you chop it into smaller pieces, now obviously you're going to have more than one single genome in a prep. You're going to probably have hundreds of thousands of copies of that genome. And so what you're hoping for is fragments to be generated of different size. Let's say from one genome you get fragments like this.
Like, would they overlap too between the two?
Ah, very good. So then shearing another one, you might get something like this.
Would you just use restriction enzymes for this?
You could use restriction enzymes, but restriction enzymes would always cut at the same place.
Right.
So you need something a little bit more random. Using something like your favorite instrument, a sonicator, actually we can shear or cut down DNA to certain sizes at random. It's a range of sizes. Typically, for this technique, it's 3 to 5 kb. But the goal would be is that you get enough sequences and enough overlap that you're looking for a certain amount of coverage. And usually they report this in terms of numbers. So like, we've read every single nucleotide 100 times. That's called 100 times coverage. Okay? And so you're looking for numerous reads, numerous sequences of one fragment or one nucleotide, so that an aggregate of all the 100 times it's read, it's like, hey, 99 times it was a C, 1 time it was a G, so we're just going to call it a C for the sake of it, right? So it's kind of a little bit of like self-control as well and built in there. Now to put it all back together, that's where the computers come in. So computers will start to, almost like a puzzle, they'll start to look for pieces that have similar sequence reads and then paste it all back together. And so if you look at this on a computer, what you'll find are fragments of different length all kind of like starting to overlap with each other. Basically, you're going to get a map that looks like this, where you're looking at each read and it's showing like, here's where they overlap with each other. That's basically what's being done. We're not going to do that. I just don't want it requires a certain amount of computing power to do it, and it requires a certain amount of understanding computer programming to do it, and we just don't have the time to do it. So what I've asked is that they just give us the fully assembled genome, we'll annotate it on the back end. That we can do without having any additional computer skills. So to give you one example then, here would be, and this is not what you would ever see, but this is an example readout of what's happening over a period of time. Each new blip represents a new nucleotide. So in this example here, you've got a blip G, and then at 105 seconds you have a C and a T, and then at 105 and 5 seconds you've got a C. 105.75 you've got a G, and so on and so forth. So within this time period right here, from 104 to 106 seconds, you've already added and read 5 nucleotides. Imagine this happening in 3 million different wells. In other words, you've read 15 million base pairs in a second and a half, roughly. So this type of sequencing gets grouped together with what we call next-gen sequencing. High throughput allows for a lot of sequencing information in a short amount of time. What we need to be able to do this type of stuff is a lot of computing power, something that can capture all of this information and make sense of it. So if anybody's looking for a second degree, bioinformatics and computer science isn't a bad degree.
Does the intensity of the fluorescence matter? Because like the first C and the second C
Yeah.
are not different.
It doesn't in this technique. It doesn't. It almost seems like, now it might be that there's a stall right here for some reason, like trouble reading the base pair. I don't know. And I took this from an example where they were trying to explain, like, what does this mean versus this, and I was like, I don't care enough to understand it right now. There are some techniques where, like, if you look at the intensity, that will tell you how many nucleotides in a row were the same. So, like, if you have a G with, like, 3 times the amount of light being released, that tells you there were 3 Gs in a row put. But again, that's a different technique, which is known as pyrosequencing. Pyro coming from pyrophosphate.
Is that where they detect they follow the pyrophosphate instead of the nucleotide?
Yeah. So they actually use the pyrophosphate to actually measure luciferase activity. It's a completely different technique that gets used, but the reads with pyrosequencing are usually shorter, like 300 to 400 base pairs. This PacBio, and one of the things that I like about it for our respect is that it's a little bit, if I'm not mistaken, it's a tad bit more pricey. But one of the things that we run into with bacteriophages is they have terminal redundancy. In other words, their ends are identical sequence. And we'll talk about that starting next week and why that happens. But in order to get around those redundant issues, you want to have longer reads. That helps you to resolve it a little bit better. There's another technique known as Oxford sequencing, and the reads for that one, you can get like 70 to 80,000 nucleotide reads, but the efficiency is really low, meaning every 7 nucleotides there's a wrong nucleotide put into place. So that one's not really good if you're trying to get the exact sequence. So we're choosing the middle of the road one.
What would be what would you use that one for?
That's a good question. So sometimes people will use that as kind of like a cursory map. So imagine, here's what I would imagine as, like, if you were let's say that you were on the West Coast and you were trying to travel to Madison. You wouldn't use a map of Madison to get to Madison, right? You would use a map of the US. That's what Oxford sequencing does, is it just gives you a very broad map to get you into the right vicinity, and then you would follow up with Illumina or another technique in order to get the exact sequence and know where everything is put based on this more global map.
So if you didn't know where your sequence was?
Or if you didn't even know, yeah, and the nice thing about Oxford is it will help you to identify some of those redundancies. And it tells you more likely where is everything in location to other sequences that you're going to get. So that kind of sequencing is used more for human genomes right now, just as a way, again, to get a cursory look of what's going on. And then you follow up with something more specific. Okay. Friday. Let's do some practice problems. So what I'll do is I'll grab 3 or 4 of them from just the review questions in this chapter in particular. I'll bring in one of the old exams as well, and we'll just kind of do a couple of them as a group. Friday would also be a good time if you have any questions about things that we've covered in the past couple of weeks that you want addressed. Let's try to get that covered as well. Monday, and we'll try to do that as we're doing bacteriophage stuff on Friday too. Sorry, I forgot to say that. Monday, we're going to move into talking about bacteriophages. Yay!
Yay!
Finally, viruses. All right, see y'all later.